I'm David Moraga with the University of Florida ICBR NextGen Sequencing Facility. Thank you for taking the time to join us in today's webinar, co-sponsored by the University of Florida and Illumina. This is the second of 4 events that are scheduled through November the 19th. Please visit our website to register for the remaining events if you haven't already done so. I would like now to pass the mic to Evelyn Griffin, the Illumina account manager for North Florida, who will introduce today's speaker.
Thank you, David, and thank you to the researchers at the University of Florida for attending the talk today. We also want to thank the University of Florida ICBR core facility for their continued interest in Illumina technology, as well as inviting us to speak with you today. Today's talk will be on advances in next-generation sequencing for immuno-oncology and translational cancer research. Our speaker today is the Segment Marketing Manager at Illumina for Cancer Research. And I want to join you with thanking Jonathan Bibliowicz for joining us today. Thank you, Jonathan, and appreciate your time. Before we get started, I just want to acknowledge a little bit of housekeeping. We will be muting everyone upon arrival, and if you could, please enter any questions that you have in the chat. We'll get to your questions as quickly as we can. As your account manager, I'm happy to answer the questions that I know the answer to, but as we advance into the talk, Jonathan has asked me to interrupt him with your questions. We will also have a Q&A session at the end of the 40-minute talk to address your questions there as well. So with that, thank you so much, Jonathan.
Thank you, Evelyn. Hi, everyone, and thanks for joining us. So there's a lot to discuss. And again, I think this is really a platform for us to have a discussion, to chat about what you're doing and where you want to go with your project. So again, please interrupt at any point. Any question, any comment is helpful for all of us. So that is really the goal here. I'm happy to stop my talk at any time. I'm going to stop my video simply to help the bandwidth. All right. So again, my name is Jonathan Bibliowicz. I manage the cancer research segment within Illumina, and I work very closely with Evelyn as well. And we're going to talk about next generation sequencing, some of the applications that many of you are familiar with, and how we're really moving towards a world of really understanding cancer in really the biology of cancer, which is really complex at different levels of biology. And I'll talk to that in a second. And it's really a question of we call multi-omics. So this is a figure from a recent Nature article that we co-authored with Nature, and it really speaks to the advancements that we've made. So in the last few years, next generation sequencing has really advanced beyond just looking at the mutations that drive cancer. Cancer is the disease of the genome, but also the changes in gene expression and things like post-translational modifications that are important for us to understand cancer. So detecting all of these hallmarks of cancer in the genome, epigenome, transcriptome, and proteome is really giving us more data to understand this family of diseases. So in addition, our ability now, and this is, I think, where we are with technology, is to combine these modes of information, we call them omics, and really adding to our ability to identify novel drug targets and look for novel biomarkers with the ultimate goal of really improving patient outcomes. That is the goal of Illumina, and I'm sure it's the goal of many of you. So in this presentation, we'll review some of the progress, recent progress enabled by next generation sequencing, and really highlight the applications and really the complete end-to-end workflows that Illumina provides, and some of them that we provide with our partners. We'll talk about single cell sequencing. We'll talk about spatial sequencing, which I think is a very hot topic, really globally. And I want to make a point that while we're going to draw from examples in immuno-oncology, which I think is really a fascinating field, these methods really work across basic and translational research, especially when you talk about cancer. So So again, the goal of really cancer research ultimately is twofold: finding novel drug targets, really improving patient outcomes with respect to therapy, and then also looking for biomarkers, whether those are prognostic, predictive, or diagnostic biomarkers. So this figure, those of you working in immuno-oncology probably very familiar with, but I thought I'll start here. It really demonstrates the immune response to tumor antigens and really highlights the role of the antigens themselves, as they play a critical role in the recruitment of T cells targeting the tumor. So everything really starts with the release of cancer antigens, either from cell death or through presentation on the surface of cancer cells. And these cause the initiation of a cycle that eventually leads to an infiltration of the tumor with antigen-specific T cells that then hone in on the cancer and hopefully destroy it. So assuming nothing gets in the way of these T cells, it's possible to assume that they alone can eliminate the tumor without other intervention, but we know that that is not quite true, and we'll get to that on the next slide. There have been several classes of therapies, as many of you know, that address overcoming the barriers of the immune system to attack and kill cancer, such as vaccines, such as antibodies, anti-CTLA-4, anti-PD-1, and then cell therapies such as CAR Ts and TILs. And all of those are just example of really the breakthroughs that we've had in the last 3 years. But all of us recognize also that there's some limitations and there's some challenges. So immuno-oncology is really the future of oncology in many ways, but there's still many challenges that we think next generation sequencing can partially help solve. So one of the main challenges are response rates. So we know that response rates in patients vary between methods and across tumor types. And again, not all patients respond at the same level. So as this example here shows, in a recent clinical trials of PD-1, so an anti-PD-1 checkpoint inhibitor in recurrent glioblastoma, only about 8% of the patients demonstrated objective responses. In many of the cancers, such as glioblastoma, which is a very challenging cancer as a solid tumor and a brain tumor, the mechanistic basis for this variation really remains unexplained. And I think that's where technology kicks in, and that's where our efforts really kick in. So again, if we revisit the figure that I've already showed, it really sort of highlights not only where cancer research is going. So really now we're looking at the disease of the genome from the tumor side, neoantigens, mutations, the traditional methodologies of next generation sequencing around DNA sequencing, really combined with methods that were traditionally reserved for cell biologists, for immunologists, such as RNA-Seq, such as things like protein detection, which we'll talk about at the very end, via sequencing, things that really look at phenotypes of cells rather than genotypes. So we're really combining methods that look at genotype to phenotype within the tumor itself, and then the phenotype of the immune cells or other cells that are within the tumor microenvironment. So again, many questions that are outlined here of what some of you might be asking regarding neoantigens, mutation burden, antigen presentation on the tumor side, and then things like T cell responses such as T cell exhaustion, infiltration, and really just the complex environment of the tumor microenvironment. And we'll get to that when we get to the section of single cell sequencing and spatial sequencing. But really, this is where science is going, and this is where technology is allowing us to go. So before I start with some examples from publications, really want to show what we offer. So again, we are a sequencing company, and you can see at the center, we have these two sequencers, the NovaSeq, which is kind of a very production scale, very low cost per sample sequencer, and then our new NextSeq 2000, which is a more accessible version of the same technology. But really the point here is that for cancer research, these platforms, these readouts, these NGS readouts really provide us to do science at different resolutions. So whether you're doing things at the bulk level, single cell, or spatial. All of that is sequenceable. And also we can look at different modes of information, right? So we talked about DNA-Seq, we talked about RNA-Seq, or we will talk about RNA-Seq, epigenetics, and now protein. So now we can look at all these omics and either do them individually, which most of us do, or really take the next step and combining these data sets, but having one readout, one technology that does all of this together or separately, I think has a lot of value and definitely for things like data consistency, but also for really starting to think about combining data sets. So we can talk about that in a second a little more. So again, one of the goals, or the two goals of in immunotherapy specifically, but cancer research in general, is really finding drug targets and finding biomarkers. So let's look at the literature a little bit and go over that. And again, if you have any questions, comments, experiences to share, please stop me, please interrupt. So the core application these days for cancer research, which has really changed over the years because we really looked at the DNA before, now it's much more of RNA sequencing is RNA-Seq. So what does RNA-Seq do? Many of you have used RNA-Seq, but I really want to outline the different types of information we get from RNA-Seq, because I think many of us don't quite realize how much information is hidden within one data set. So RNA-Seq really reveals the key components of the anti-tumor immune response. So we can look at, as you can see in blue, immune infiltrates. So we can use deconvolution algorithms from bulk samples to really see what cell types are within the microenvironment. So identifying cell types and gene expression changes of the microenvironment. We can also look at expression phenotypes. So we can identify first, again, what cells are present, but also through expression signatures, really understand what these cells might be doing. And maybe we might have some actionable targets that we can work with. And also we can look at repertoires, things like TCR sequencing and BCR sequencing to really identify immune cells at a more clonal level. And we'll get to that in a second as well. So here's an example from a recent paper from Nature. This was, I believe, out of MD Anderson. And really what they wanted to look at in the context of this specific cancer type, and this is in melanoma, to really assess immune infiltration composition. So really look what cell types, immune cell types are present within the tumor microenvironment, and maybe those will be actionable targets for interference or biomarkers. And most of the field has really been focusing on T cells. So T cells are kind of the workhorses of immuno-oncology, but in this specific context, bulk RNA-Seq really identified B cells as sort of a novel cell type that might be active in these melanoma tumors. So again, just showcasing how we can really start to understand the infiltration of the tumor microenvironment, even with bulk samples that don't have the resolution of more newer technologies like single cell, which again, we'll talk about in a second. So by identifying B cells, we now have a biomarker for patient response. So you can see the graph on the bottom right of overall survival probabilities that correlates with the presence of B cells, but also we can start understanding what are their function and how we might improve their ability to do their work and to attack and kill the cancer by really looking at the transcriptome, really looking at the expression and the subtypes of B cells within the tumor. So again, in addition to identifying cell types, RNA-Seq can also use the data to generate the full transcriptome, and again, begin to understand cellular phenotypes, the states of the cells and the cell function, which I think is probably the most critical. So this really allows for a really deep understanding of mechanisms and really serve as in the identification of drug targets. This next example is a really nice example of RNA sequencing in drug target discovery. It comes from an Australian group and published recently, late last year, in Science Translational Medicine. They used RNA-Seq to try to understand the biological mechanisms that underlie response to immunotherapy. This is with the idea that having a better understanding of these mechanisms could identify pathways that can then be targeted in combination with immunotherapies. So they looked at, as you can see on the slide, differentially expressed genes in responder and non-responder first mice models, but then in actual patients, after double treatment with 2 immunotherapies, anti-CTLA-4 and anti-PD-1. So really trying to compare, are there any differences between responders and non-responders? Can we learn anything about biomarkers? And can we learn anything about actually improving the response rates? So again, this was done in mice and then later on in patients. So their RNA sequencing, and again, this is bulk RNA sequencing, identified 3 pathways. So as you can see on the slide, STAT1, interferon gamma, and IL-10 as pathways that might underlie differential drug responses. So these pathways were drug targets, putative drug targets. So when they actually targeted these pathways with drugs, with a drug combination, with a new cocktail, together with the anti-CTLA-4 and anti-PD-1, they were able to increase the baseline or the response rate from 10% to really, I would consider an astonishing 80%. So really the 10% in some of the cases that we saw before response rate could be increased to 80% based on just new findings from genomics to really understand what pathways are dysregulated, activated, and such, and really try to increase those percentages substantially, as in this case. So this is, I think, a really beautiful example of this. So what does this study really show us? It really supports the validation of these pathways as drug targets in combination with existing drugs. This could obviously be done in a context of where no drug is used as a standalone, but also found biomarkers for patient selection. So really, within the same data sets, we're getting those 2 data sets, these 2 insights, biomarkers and drug targets. And it really supports, I think, the use of RNA-Seq in discovery, in immuno-oncology and beyond. And I think the scientific community is realizing that, and we're seeing more and more adoption of RNA-Seq in this context. So from the Illumina side, you know, when you think of RNA-Seq, some of you have done it. Some of you might think it's a bit scary, but I think as a company, we've done a good job, especially recently, of really creating what we call comprehensive workflows, just making it simple. I remember in graduate school, I was very afraid of this. This was, 12 years ago, but now I think the experience of running an RNA-Seq experiment is much more seamless. There's a clear end-to-end solution, anything from actually getting your samples, extracting the nucleic acid, library prep solutions, which your core can help you with. The sequencing is really simple and straightforward these days. And then we have built-in and data analysis platforms that you can use. So really, for researchers, especially going through the core, this has become very routine and very easy. And as hopefully through the previous 10 or so minutes and in the rest of the presentation, I'll convince you that if you're not convinced already, that the insights gained by sort of discovery approaches, looking at the whole transcriptome, looking at the whole epigenome, and really layering all these levels of information is just critical. It's almost needed these days to really understand, get new insights into cancer biology. If you're curious about other workflows beyond RNA-Seq, we have those and we're happy to share those. So again, just kind of ending the RNA-Seq section, if you are interested in certain types of RNA data, such as just mRNAs, just looking at the expressed exome, there are solutions for that, from extraction all the way to library prep, and then obviously the sequencing. On the right side, if you're looking at total RNA, so if you're looking at things beyond the mRNA, like non-coding RNAs and smaller RNAs, you can do the total RNA. And then if you have something like an FFPE sample or something that's a bit more degraded, we can use an enrichment protocol to really pull down and isolate the fragments and get very high quality sequencing data using that approach. So the information you want to gain from it, or the sample type, poses very few challenges these days because we have this menu of prep solutions. And I think an important point, as someone that has done a lot of qPCR in the past and panels, as we know, there are many panels on the market. So, if you're working on immuno-oncology, different companies provide different panels that look at dozens to hundreds, sometimes thousands of genes. And that's great in many contexts. But what this figure shows is that whenever you use any panel, any choice, predetermined choice of genes that you want to look at, whether this is on the DNA or RNA level, in this case, that this is RNA-Seq, when you compare the different panels, for example, for immuno-oncology, from NanoString, or even from us, I have a couple of our own products here, there's very poor overlap between the panels. So, you're really limited by the content that you provided. And again, in some cases, this is very useful, but as the bottom bullet point highlights, this really underscores the need to perform hypothesis-free full transcriptome sequencing, really true discovery that is now not only more accessible these days, but is just not limiting. It's just you really looking at things, really doing true discovery in research. And really quickly, RNA-Seq is actually providing more sensitive information. So you're picking up more of the transcripts that are, especially the low expressing ones, if you're looking at things like transcription factors and things that are low expressed in your sample, then it does as good of a job, and it's sometimes much better than panels, which is a bit counterintuitive, but all of the data shows that is true. So you're getting, again, the full scope of the transcriptome, plus you're picking up a higher percentage of your RNAs, especially if you're looking at the low expressing ones, which most of us are. So sensitivity is also has been tremendous with some of the new products. And just in general, there are many technologies out there for RNA sequencing. This is a paper that has been updated since. This is from 2016, but this is out of the Journal of Immunotherapy of Cancer, where a panel out of SITC really evaluated different technologies for genomics, especially for RNA-Seq, and really came to these conclusions that are outlined here graphically of really next generation sequencing, really providing, as you can see on the fourth line, really the richness of information. So just discovery power, which no other technology does, but also as far as the technical aspects, the sensitivity, the multiplexing, and some biases like PCR biases, as far as data quality is more robust. So across the board, if you can read the quote on the bottom, really for immuno-oncology, next generation sequencing has and is becoming more and more, because of the capabilities that I have showed you and will continue to show you over the next 10 or so minutes, the platform of choice for immuno-oncology, because we can do so much on with one technology. All right, so I imagine that what I've said so far is a review to many of you, but I'm going to slow down a little bit here. There are two angles to RNA-Seq and to really cancer multiomics that have really revolutionized what we do and how we do it. The first is single cell sequencing, and here I imagine that, again, there are some of you that have done it and some that might be still curious. This figure that I'll use to explain single cell sequencing is taken from a recent Nature Medicine review. I definitely recommend this review. This reviews the power of single cell sequencing in immuno-oncology specifically. So the discovery power of RNA-Seq can now be extended to single cell resolution. So the tumor microenvironment, as you can see on the left, is composed of really various immune cell types and non-immune cell types. So you can now take a tumor sample, or any sample for that matter, dissociate it and sequence each cell individually. What this allows you to do is to pair the identity of each cell, which we did previously with bulk RNA-Seq, or each cell population, with its phenotype, function, and state. So now you're doing that for every single cell, and then compiling the data and understanding globally the composition, states, and function of subtypes of cell. So the different cell types not only are present at different proportions, and we saw that with the B cell example early on, but also they respond to therapy differentially. So that is on the right side of the figure. So to really understand response to a drug, response to a treatment, respond to a mutation, response to any challenge that we give the tumor or its microenvironment, we really need to understand the differential responses of the different cell types. So by analyzing the tumor endomicroenvironment, let's say before and after treatment, before and after immunotherapy, we can really better understand how the cells respond, identify novel drug targets, which are usually cell type specific, and really design even more effective and more personalized combination of existing drugs. So this is where the field is going, and I hope that this slide and the next would convince you if you're not convinced already. But this really is allowing us to understand a very complex system and give justice to its complexity, and really target drugs to where they need to be targeted, and understand if there are any side effects with response to the treatment of other cell types. So one thing I think that is really revolutionizing genomics, as I mentioned before, is the ability to simultaneously look at the transcriptome, the full transcriptome of each single cell or in bulk, and also look at protein detection. And this example, this figure is taken from a technology that is now commonly used, and I believe that your core is well versed in this, called CytSeq. And CytSeq technology is basically antibody detection, just as you would do in immunohistochemistry or a lot of the protein detection methods. But now, instead of connecting the antibody to a fluorophore or a reporter, it is conjugated to an oligo. This oligo really imitates mRNA. You see the poly A tail on the right. So when you expose the cell preparation that you've prepared for single cell analysis with these antibodies, you're basically pulling everything together, and you can sequence all of the RNAs within a specific cell, plus up to dozens and now over 100 antibodies. So what this allows you not to only understand the complexity of biology across, protein and RNA, but also to identify populations based on known markers. Most of the markers that we use in immunology and sometimes in cancer are protein-based, CD4, CD8, etc. Now you can identify those based on your markers of interest, if you come from the cytoproteomics, and then dig in to the transcriptome and do all of the work that we describe, biomarkers and drug targets. So I think this is really, really revolutionizing cancer research and really connecting the worlds of traditional cancer research with the new angle of immunology for immuno-oncology. So multi-omics, this is one example of really multi-omics, really looking at different levels to connect data sets to understand biology at a deeper level. But since cancer biology is complex, multi-omics also allows us to find clues that underlie the causes of things like initiation, progression, and relapse of cancer. So in this paper that came out, I believe last year, or 2018, this is from the Fred Hutch Cancer Center in Seattle. This lab researched the underlying reasons for a specific type, for relapse of a specific type of cancer called Merkel cell carcinoma in patients treated with virus-specific T cells plus anti-PD-1 checkpoint inhibition. So this is, again, a cancer caused by a virus. And they had existing therapy that combines CD8 T cells as an immunotherapy plus anti-PD-1 inhibitors. As you can see on the left, there was an initial response in the patients, but after about 32 months, there was a new metastasis and there was a relapse. So the core of the question in this paper is why do the patients relapse? So over the course of the beginning of the research, they looked at different methods. They did things like H&E staining, this is on the right, immunohistochemistry for the antigen itself to see maybe the relapse is a result of the antigen not being presented to the T cells that are being used. They looked at the MHC class, which is really the molecule that presents the antigen to the immune cell, nothing there. They actually did whole exome sequencing, looking at maybe there's a mutation of the virus itself that sort of evolved and now evading the immune system. There was nothing there. They also used Sanger and other methods, and there are really no clues at this point to why the patients are relapsing. So this was just at the time where the company called 10x Genomics came into the picture, and actually the first publications out of 10x Genomics in collaboration were done at the Fred Hutch, and this group decided to use single cell sequencing to look at really high resolution transcriptomic signatures in these patients. So as you can see on the left, around the time of relapse, there is an expansion of CD8. So these are, if you haven't seen these are t-SNE plots. This is single cell data where you're getting the immune cells from the tumor, the microenvironment, or from the blood, and each color scheme represents a subpopulation of cells. So each dot is a single cell, based on the full transcriptome of the single cell that identifies the cell, its identity, and its action. And then you sort of combine these plots together to look at what cells are present, and then start to dig into specific subpopulations and understand the transcriptome and their function. So they want to see if there's anything abnormal that's happening again at the time of relapse. So looking more closely at the transcriptome, the single cell level, before and after therapy, they found a loss of the HLA gene. So the HLA gene codes for the MHC gene, which you saw earlier, and is responsible for the presentation of the viral antigen. So this loss of HLA gene transcriptionally, so we know of loss of HLA on the DNA level, but transcriptional loss is really, was at the time, a novel mechanism for tumor invasion. And indeed, as you can see on the right side of the slide, it was further found that HLA loss was reversible. So you can actually use pharmacological doses of either interferon gamma or hypomethylation agents to reverse this transcriptional regulation of HLA and overcome the relapse. There are other sort of details that are really interesting. On the very bottom right, you can see that two of the patients, one had loss of HLA-A, one had loss of HLA-B. So there's some nuances to which allele or which gene is actually being transcriptionally regulated. But again, just showcasing how the resolution of single cell in the context of RNA-Seq really helps us understand novel mechanisms, and really looking at all the different levels of biology, the DNA, the epigenetics, the RNA, and protein. We just don't know where the misregulation is. So we really ideally should look at all these levels of biology. Excuse me, I'm going to skip one thing just because of time. But for those of you that have done single cell sequencing, and those of you that haven't, it's a confusing world, and we acknowledge that. There are many, many different ways to prepare tissues, so different companies that have solutions to do that. There are different ways to isolate the cells. So I mentioned 10x Genomics, but that is one of many platforms that are available. Sequencing is fairly straightforward, and it's mostly Illumina sequencing, and that is probably the simplest part of the workflow as far as choice. And then there are, different companies that do the data visualization, which actually these days is actually fairly standard and integrated into the solution. But on the left side, there are a lot of choices to be made. And I think the core facility at the University of Florida has made a lot of the choices for you. But if you have some more questions, we have what's called an ebook, a single cell ebook, and we're happy to share that as a follow-up to the seminar. And that really outlines the different considerations going into a single cell experiment. So again, from the tissue preparation to the isolation, the sequencing, and the analysis. And I think that is a really useful resource, especially for those of you that are more novice. Okay, I'll pause here for a second. Any comments or questions?
Nothing in the chat, but
Okay.
you can feel free to unmute yourself very quickly and ask any questions.
No? Okay, we'll move on to, and I'll really slow down here. This is, I think, probably the most exciting part of what is going on in technology is spatial transcriptomics. So for those of you that have heard or haven't heard about spatial transcriptomics, It's a fairly new technology in its current form. And really, it's about bringing together the worlds of histology and sequencing. So really allowing us to study the tumor and the microenvironment. And again, this is especially powerful in oncology and cancer research. And this is done in situ, so it's done on a slide to really understand, again, the complexities. So some questions that we ask from single cell, we can now do the same analyses or very similar to it on a tissue section, on a slide, to really look at architecture, to really look at how cell types relate to each other spatially, and really is, I think, helping people that are more used to imaging, really looking at solid tumors, to really enter the world of next generation sequencing. So basically what it does is there are different technologies out there, but all of them really do the same thing, which is really getting your section, whether it's FFPE section or a fresh frozen section from a solid tissue, and first imaging, determining what area of the section you want to look at, and then extracting those cells of interest and performing whole transcriptome sequencing on, with single cell resolution. So it's really taking single cell again and adding another dimension, which is the spatial context of the tumor. And those of you working in cancer, I don't have to explain how important that is in oncology. So the figure on the right is taken from, again, another Nature review that we did with Nature that just came out actually last week. Outlines the differences, the type of insights that you gain from bulk sequencing on the left, which is really about, as the example at the beginning, really understanding in very general terms, what are the contributing cell types within the tumor, what are the general expression changes or genomic changes or epigenomic changes within the tumor, or comparing tumor and normal. But what we're missing there is real resolution, really understanding what cell types are present with accuracy. And that's what single cell does for us. So single cell is, again, dissociating the tumor, sequencing each cell individually, and then grouping them into cell subtypes and groups, and understanding their phenotypes and their mode of action within the tumor and its microenvironment. So it has just more resolution. And then again, we're taking that to another level with solid tumors and really looking at that same information, full transcriptome, and often together with protein information. So full transcriptome plus dozens of proteins on one section. So this is, again, truly revolutionary because as compared to the other methods out there like immunohistochemistry, in situ hybridization, that really look at just a few parameters, now you're doing true discovery on a slide. And that is really, really, really exciting for all of us. So just to, again, see how we're doing on time, but a quick example, I think a simple example for, why we need spatial information, and then we'll expand on it. On the left, you have just a section of what we call the cold tumor. So this is a tumor where there's no infiltration of immune cells, and you can see the absence of this brown precipitate that is often used to detect these immune cells. In the middle, you have what we call a hot tumor. So this in Immuno-Oncology just means immune cells are present in the tumor. So the cells are in there. We don't know what they're doing because we're just detecting them. And that's where things like RNA sequencing will come in, saying what are the functions, what are their true identity, and are there any blocks that we need to remove in them actually attacking and killing the tumor. But we also have situations where if you do a bulk analysis or even single cell analysis that on this tissue on the right, you will detect the brown precipitate. You will detect the T cells that are present in the tumor. But when you actually look at the architecture of the tumor, you see that those immune cells, whatever their identity is, we don't know based on this assay, are actually on the margin of the tumor. They're actually not infiltrating. So with traditional methods in the context of oncology, we're getting partial answers that actually sometimes deceiving. So the power of spatial analysis is to really understand the interaction of cells, their architecture, and how they, in addition to understanding the phenotype and the full transcriptome of the cells. So hopefully this example helps. And I'm happy to answer any more questions about spatial genomics. It's, again, a fascinating field. I'm going to end with going back to the original paper that we showed, which is the B-cell paper. So this is, again, out of MD Anderson. This is in melanoma. And in this paper, they started to, and this is from Nature from beginning of this year, use spatial, in this case, proteomics and transcriptomics, and compare that to traditional methods such as immunohistochemistry. So the goal here is to understand what cell types, again, are within this melanoma tumors and understand a bit more about what they do. The initial discovery was that B cells are really, can really distinguish responders and non-responders to immunotherapies. And then they wanted to look more closely at different cell types within the tumor. On the left, what you have is some just images that many of you are familiar with of traditional immunohistochemical images. So you have a section, you have maybe H&E staining, and you have, some sort of either a fluorophore that is connected to antibody or this precipitate reaction. They're really looking at one protein at a time. Or in the case of in situ hybridization, be one gene at a time, or maybe you can layer a few of them on top of another, but that is extremely challenging. On the right, you start to see what you get with spatial analysis. These are actually protein markers, but again, you can do protein markers together with RNA and combine that all on a sequencing run. So both the protein and the RNA are sequenceable together. But what you can do on the right side is start to look at multiple parameters at the same time. And if you have the full transcriptome on the same section, you can pick and choose with your software which ones you want to look at. So you can use, look at a couple of proteins together with a few of your RNAs of interest and really start to go through the full transcriptome and visualize it on the section. And to me, doing a lot of immunohistochemistry and in situ hybridization years ago, this would have been just phenomenal to really, again, identify cell types using normal markers or known markers, but then truly understanding what these cells do. And if there are any dysregulations on the RNA level that might explain certain phenomena or phenotypes, such as resistance, such as response, such as biomarkers. So hopefully you're as impressed as I am, as excited as I am. But I think this is where cancer research is really going together with single cell. I think single cell will continue to have an impact because that not only single cell is sufficient, but actually, so we really have to make decisions about which technology we're using to answer our scientific question, our research question. So I think I'm out of time, but really want to end this here again and really make the point again that we believe, and I think the scientific community is starting to realize just the power of genomics. Genomics has moved away from just DNA sequencing, whole genome, and looking at mutations. It's now a readout for so much. We can do things like methylation, like RNA sequencing of all types of RNAs. We can do protein. We can do metagenomics. We can look at microbiome in cancer patients, and all of that is sequenceable. Not only is it sequenceable, it's sequencing on the same run. So you can collect all of this data for your research and sequence it all together in one single run. And if you're really adventurous, we get into the point where you can start combining data sets. You can start to really look at very complex biology at the genomic, transcriptomic, epigenomic, and proteomic level. So I'll stop here. Again, thank you for joining us. I feel like I'm going a little fast because we're short on time, but hopefully this was clear. And please, if you want to unmute yourself or just send a question or a comment in the chat, would really appreciate it.
Thank you so much, Jonathan. While people are sort of digesting and thinking about their questions, one question that I had was just in general, I'm not sure if you know the answer to this, the sequencing requirements for a single cell versus spatial. I know that they have like recommendations on a certain micron size of the in situ piece of the slide that you're pulling the cells from. Do you have a feeling for that, or is that something we would follow up with on the given?
I can provide a general answer. Yeah. So right now, there are two sort of leaders in the field, and that is 10x Genomics. They have what's called the Visium platform, and then there's NanoString, which has their Geomics platform. They're similar yet different. So just in a nutshell, the way the Visium platform works from 10x Genomics is really you have certain areas that is almost like a microarray. So basically you put your section on this slide, and then you're really sequencing everything, right? You're sequencing the whole slide. So that usually requires on the average, and I'm giving you a rough number, I'm not sure if it's 100%, but around 200 to 400 million reads per sample. So that is on the higher end. The NanoString platform is really, you can choose your regions of interest, which I think is just has different applications. So if you're imaging, you're looking at the slide, and there's a region of interest of areas, and that's based on fluorescent, right? So you attaching this fluorophores either to the protein and RNA, so you can image before you sequence, you can image based on protein or RNA markers. Once you understand which part of the section of the slide you want to actually sequence, you actually mark those areas, and I believe you can have up to 12 per slide, and only those are sort of sucked up by the instrument and analyzed. So that my point is that because you have a region of interest, the sequencing requirements are lower. So we're talking more around 50 million reads. So you can do really on a really standard sequencing run. Now what we do is about 8 samples per run. So 8 samples, each one has 12 regions of interest. So 8 slides basically. That is sort of the standard of what we do with NanoString these days. So it really depends on the scientific questions, what sort of information you want. In fields where you want to look at everything, you sort of atlasing your tissue. You just want to sequence everything, then maybe the Visium platform would be better. I think in oncology, there's some real strengths to the NanoString system, but again, it really depends on what you're looking for. So that we can follow up with more consultation, but that is kind of the general answer. But again, multiple slides can be analyzed as the NanoString platform, which we have a full workflow going from their preparation for Spatial into the NextSeq 2000 specifically. On the NextSeq 2000, you can do 8 slides on a single run or more. So that is sort of the minimum.
Thank you.
John, this is David here in the lab. Can you comment, make a few comments, if you would, on the single cell preparation of single cells step, right? The upstream part of the whole workflow. I think that at a practical level, I know for a fact that a lot of people struggle with getting their samples into single cell form. Of course, and you have solid, it must be easier in certain situations, but not so much so in the case of solid tumors, for example, and so on. And also, if you can comment on the effect of that whole process of singularizing the cells and the potential effect that it may have on the biology you're trying to understand. How do you avoid that?
Yeah, those are great questions. So as far as the prep, I think that's why we put together the single cell ebook that I showed you, and we're happy to share that because of the challenges. Usually there are certain, I believe you work with 10x Genomics, I think that they have application scientists and people that can help with that. But really the preparation is really standard. I mean, all of these platforms accept sort of the same input, right? So you need to dissociate. And there are challenges around that. And it really depends. There's no one answer because, there are tissues such as like people working on brain tumors, for example, with all the dendrites and the neurons, that it's really hard to dissociate, that they actually extract just the nuclei, and you can perform nuclear seq on the nuclei and get a good representation of the cytoplasmic RNAs. There are so many different ways to approach a problem that I think it just requires a more individualized consultation of what's your sample type, how you're dissociating, and what information you're trying to get. So there's no overarching answer. That's why, we created this ebook. And because it is actually complex. So we're happy to help, Evelyn and the team. We have a lot of expertise, so help with that. But if you work with 10x Genomics, I suggest contacting them first. The second part of your question, sorry, was around.
Yeah, the concern that the handling of the cells that you're trying to understand, that you're studying, the concern that all this handling, the whole process of teasing the cells out into single cells, the tissue out into single cells, and so on, whether it's centrifugation, putting them through high Gs in a flow and things of that sort. But how do you minimize the impact of all this handling on the biology that you're trying to understand?
So very early in the field of single cell, and I used to work for a company called Fluidigm that was the first company that started doing single cell. And we had a lot of pushback on this, but all of the publications that came out at the very beginning, and I think that question is fairly settled. That there's no real effect unless you really keeping your cells in inappropriate temperatures and conditions for a very long period of time. So the way they do this is they do these comparisons where they take single cell data, they consolidate and do what's called like a pseudo bulk. So basically combining the transcriptomes of all the cells and seeing if the transcriptional profiles fit bulk analysis. And the correlations between the pseudo bulk, which is again, taking all the single cell data together, and the bulk have been, are phenomenal from what I remember. And I can share those publications with you. So I think the conclusion in the field in general is that if you follow the protocols of things like 10x Genomics or Fluidigm, whatever, there are no major changes in transcriptomes that really would affect your conclusions. There are some in rare cases of like the cells are lysing or dying. There's some, I think it's mitochondrial genes or things that are really show stress to the cells. But then if you identify those, then you can exclude those from the analysis. But my understanding and my memory tells me that is a sort of a rare situation. So there are filters that you can put your cells through and say, I'm seeing cell death genes, or I'm seeing stress genes, and I would like to exclude these specific cells from the analysis.
Thank you.
Sure. I'm curious about, if people are still on the line, about spatial biology, if there are any questions there as far as the value of it, and what sort of questions or projects could be done using spatial.
Nothing in the chat, but
okay.
if anyone feels free to unmute themselves. Jonathan, I've seen a slide in the past where, comparing it was just a nice visual. It was like, transcriptome was kind of a fruit smoothie.
Yeah.
And then single cell was like the fruit salad. So you could see the blueberries from the strawberries, from the, watermelon. And then spatial was this, because it was like a tart where you could see where they were in relationship to each other, obviously. And so for me, it feels like in a tumor environment, the relationship between these different cell types and how the tumor responds to treatment is crucial, right? And the only thing I struggle with is how this translates into the clinic, right? So it basically, I think, helps with like drug choice, response, and things like that. But that takes a long time to translate into the clinic. My question is, how does spatial help that transition to happen quicker or happen faster?
That's a good question. It's something, I can speculate a little bit. And I think on the research side, the basic research, there's just an immediate and obvious benefit for using spatial in some cases, right? Such as solid tumors, where context matters. Spatial context architecture matters. So that's clear. What we're seeing with single cell is, I won't say clinical, definitely not yet in most cases, but really taking clinical trial data samples and using single cell. And there are a couple of publications, recent publications that are doing that more and more, understanding patient responses using single cell analysis. So I think in a clinical trial, phase 1, phase 2, we're seeing more and more incorporation of single cell with kind of just a research angle. And I see no reason why spatial would not, it's going to take a bit of time as far as adoption and, but spatial is basically single cell on a slide. I mean, the technology is so good that even with some researchers these days that are, lost my train of thought, that are doing liquid, so like blood samples, are actually just putting their liquid, their cells or cell culture on a slide and doing spatial. They're not getting spatial architecture, but the workflows are so easy that, if they have that, they can do it on solid or liquid tissue. So again, these sort of like end-to-end workflows that, we work on, whether it's for bulk, single cell, they're just so seamless these days. And the bioinformatics are really, for like nanostring or for 10x, the bioinformatics are there and fairly user-friendly that there's no reason not to do it these days. The adoption in clinical setting is going to take time. I agree with you. But we're seeing this more and more in clinical trial settings. And I think, again, spatial is going to make that transition fairly quickly. Spatial has another angle, which is pathology, right? And that's something that if any of you are going to the Association for Molecular Pathology conference, which is at the end, that's called AMP, at the end of the month, we are doing a session on spatial genomics and really having a panel discussion with pathologists around this topic, really exploring the fit of spatial genomics in that context. So that's one case where I think spatial could fit in the short to medium term.
Yeah, it's really interesting.
Yeah. So if you're at AMP, we're going to have a workshop together with NanoString, and we'll have pathologists. We won't speak much. We'll just have them brainstorm about exactly the question that you just asked.
Right. Well, we are at the top of the hour, actually. Still nothing in the chat, Jonathan, but you covered everything beautifully. Thank you so much. And
thank you all.
We did get one question in the chat about sharing the slides and the recording. And so I'll follow up with Chris and David with the ICBR core in regards to that question. And David, you'll enjoy this. There was also a question on a certificate of training. So I'll follow up with you on that as well. But thank you so much, Jonathan. We really appreciate your insights on next generation sequencing for immuno-oncology and cancer research, in particular, transcriptome versus single cell versus spatial sequencing. So with that, thank you to the ICBR core facility as well, and for those of you who attended today.
Thank you.
Bye, everyone. Thank you.